So when we left off in the last lecture segment, we were looking at the molecular structure of chlorophyll, and I was pointing out that chlorophyll has a porphyrin ring, which looks like kind of like this little honeycomb structure here. And this is the part that absorbs the photon of energy, the light energy. And then it has this hydrophobic tail, which attaches it into the thylakoid membrane. So there's somewhat of a similar structure in all of the other pigments, but you can think of it in this way, inserted into the membrane, and then the part that absorbs the photon on the surface of the thylakoid membrane, which you'll remember is inside of the chloroplast. And so what happens is when the photon hits this porphyrin ring structure, it excites or takes the electrons that are there and moves them to higher energy levels. So we say the electrons become high energy electrons. And they're going to be then passed to something else, an electron carrier, another electron acceptor in this story. So really, all of these pigments are in these little groupings, in these little regions of the thylakoid membrane. And when you have hundreds of these pigment molecules kind of in a group, we call it the antenna complex. And the antenna complex involves all of these pigment molecules that are all absorbing photons and then passing that energy to the reaction center. And so it's only the reaction center, this special chlorophyll, and sometimes it's two chlorophylls, that are actually getting the photon energy from the other pigments around it. And that's the one that gets its electrons excited or energized. And those electrons are going to move out of the photosystem. So the photosystem, that term refers to that grouping of pigment molecules, and then in the center, the special chlorophyll molecules. They're called the reaction center chlorophyll. In this picture, this is how the artist of your textbook decided to draw it. I don't know if I like this drawing because it almost looks like the chlorophylls are in some kind of big bubble, which they're not. They're just laying flat on the thylakoid membrane, but we'll forgive the artist for that. And they did show the chlorophylls as green, so I guess there was something good there. But you imagine this grouping just inserted, and these are supposed to be over here. These are the hydrophilic heads of the phospholipid bilayer. So I wish they hadn't colored them. They almost look green to me as well. So they would have no color. But the chlorophylls inserted there would have the green color. And so here in this diagram, they're showing a photon hitting one of the many chlorophylls in that antenna complex, in that grouping of chlorophylls. And then they're showing with the black line that energy from that photon is passed from one pigment molecule to another, but ultimately, no matter how indirect that path is, it ends up getting passed to that reaction center chlorophyll, that special chlorophyll that's in the middle, sort of. And that molecule is the one whose electrons get excited, and then these electrons shown here are passed to an electron acceptor. And I wish they didn't show it like shooting up. The electrons aren't like shooting out of the membrane. They're just being passed to an electron acceptor. In fact, the electron acceptor is going to be right next to this photosystem. It's going to be embedded in the membrane over here, for example. So this is just the way that the artist chose to draw it. It's not that unusual, frankly. And but they're showing the path of the energy, and then the energy is then combined with the electrons in the reaction center chlorophyll molecule, and then those are being passed. Those high energy electrons are then passed to an electron acceptor nearby. So this diagram, this is a favorite of biochemists. What they like to show on the y-axis is how much energy the electrons have. Now, guys, the electrons are not shooting out of the thylakoid membrane. They're actually staying with the electrons and they're being passed down an electron transport chain that is a group of embedded proteins or transmembrane proteins embedded in the thylakoid membrane. But when you draw the picture and you indicate the level of energy that the electrons have at any point in this process, then you end up with something like this. And so I'm going to try to explain it. This is really happening along the membrane. So think of the thylakoid membrane here, and you have this group of pigments here and a reaction center right in the middle. And that reaction center, has two electrons that get excited by the photon. And then it passes those electrons down the electron transport chain along the membrane. But when you do it when you kind of graph it, this is almost like a graph with the y-axis being the energy of the electrons, then what you show is the electrons go from being low energy when they're at the reaction center to being very high energy. And then this part is showing them being passed along the membrane down the electron transport chain. And then they actually reach another photosystem nearby, and then they get activated again, and they go down another electron transport chain. So they actually get reactivated in the middle of the process. They get activated by a photon here, and then they go to another photosystem nearby, and they get reactivated by another photon there. In the end, what we want to focus on is the purpose of any electron transport chain, at least the one that you know of, is to make ATP by building up protons, by pumping protons across a membrane, creating a gradient, which is then allowed to flow back through ATP synthase to make ATP. And that is what's happening here. These electrons are being passed down the electron transport chain, and at the same time, protons are being pumped against their gradient into an area that already has a high concentration of protons or hydrogen ions. And then there's this ATP synthase, and it's exactly the same ATP synthase. It's not the same protein complexes exactly, but it is the same ATP synthase. And the whole system works for the same purpose, to make ATP. So the protons flow back through ATP synthase, it turns the turbine, and it makes ATP. And so in this diagram, they're showing you kind of this over here in a way. So ATP is made. And then, so circle that, ATP is made. And then when the electrons go down the second electron transport chain here, they get picked up by NADP, and it becomes NADPH. So you'll remember in the summary, when we were talking about the light reactions or the light dependent reactions, we said the purpose of the light reactions is to make ATP and NADPH, which are going to be needed for the second phase, which is called the Calvin cycle. So here's where that's being made, NADPH and ATP. Now, when you have a photon that hits this first reaction center, two electrons from that chlorophyll molecule are passed down this two electron transport chains, and they finally end up in NADPH, at least temporarily. But what that means is that the reaction center is now short two electrons. It needs to replace them before another photon hits. Because if another photon hits, and that energy from that photon is passed to the reaction center, there's no electrons available there to energize a second time. So what happens each time a photon hits, a molecule of water is broken apart, and two electrons are taken from that water molecule, and they're taken by that reaction center chlorophyll. That's what makes the reaction center special. It can split water. And if you think about that, it is impressive. It can split water. It can break apart a water molecule and take two electrons out. And it needs those two electrons because that photon is going to charge them. And so each time a photon hits the photosystem, a molecule of water is split to provide more electrons. And then the next time a photon hits, another molecule of water is split. And the next time a photon hits, another water molecule is split. So this keeps going on and on. And so when the water is split, that process is called photolysis. It just means splitting water with light, what it means. But that's what it means. The water is being split. It's really being split by the reaction center, but it's because of the photon hitting it that the reaction center needs more electrons. And when water is split, the electrons are what the reaction center wants, but the oxygen is released as a byproduct or a waste. And you may remember that oxygen is a product of our overall reaction summary of photosynthesis. So water is needed for photosynthesis as a source of electrons that can be charged by an energy from the sunlight, from the photon, and oxygen is a product of the breaking apart of the water molecule. The plant doesn't, for the purpose of photosynthesis, the plant doesn't see the oxygen as something good and useful, although we know that the oxygen can be used by the mitochondrion for respiration. But for the chloroplast, the oxygen is a waste. It's a waste product generated when water is split apart. So water is a source of electrons for photosynthesis. And oxygen is a waste or a byproduct or a product. And in this process, ATP is made and NADPH is made. Now you may be wondering why this photosystem is called photosystem 2 and this one's called photosystem 1. It turns out that the chlorophylls they have are slightly different. And it turns out that the one that is named photosystem 2 was discovered later than photosystem 1. So in chronological order, photosystem 1 occurs first, and in chronological order, photosystem 2 occurs first, and photosystem 1 occurs second. But in terms of how they were discovered, photosystem 1 was discovered first, and so they gave it that name, photosystem 1. And then it was like, oopsie, there's another one that actually occurs before that, but instead of changing the name, they just called this one photosystem 2. So it's kind of annoying. Okay, so just to remind you, the protons are being pumped by that electron transport chain, and the proton concentration ends up to be higher inside the thylakoid space than out in the stroma, all right, because of the protons that are pumped across the membrane by the electron transport chain protein complexes. And when water is split, that adds even more protons to the thylakoid space. All right, so that creates a really high concentration of protons inside the thylakoid space. And that is going to then be part of what's the energy that's used to make ATP, because ATP, you'll remember, has the ATP synthase has this channel in it that allows the protons to diffuse back into the stroma, and that turns that turbine, which causes the production of ATP. And that's the same thing that we see in the mitochondrion. So this diagram tries to put it all together and tries to arrange everything in the way that it would be arranged. And they're just showing one thylakoid disc, not even a whole one, a part of a thylakoid disc. You know that you have lots of thylakoid discs, so that means this setup here is, copied in all the thylakoid discs many, many times. But they're just showing you one. A photon hits this particular photo system here, the antenna complex, or all those chlorophylls, and in the center, which in this picture I don't see where their center is, there's the reaction center, and when that photon is passed to the reaction center, 2 electrons are taken from water and charged or given energy from that photon. And those electrons are then passed along the membrane, like I was saying before, till they get to the next photosystem, which is actually called photosystem 1. And then they get recharged by another photon, and eventually they get passed to NADP+, which makes it become NADPH. And as these electrons are passed down the electron transport chains, there's a pumping of protons into the thylakoid space, which causes a buildup of protons inside the thylakoid space. And there's a channel protein here, which they can diffuse back through out into the stroma. And that turns the turbine of the ATP synthase enzyme, which makes ATP. So we make ATP and we make NADPH, which is what we need for the second phase of photosynthesis. And we also make oxygen as a product of the splitting of water. Okay, now we're ready for the second phase. The second phase is the Calvin cycle. The Calvin cycle is named after Melvin Calvin. I don't know if that's a name choice I would have picked, but Melvin Calvin. And Melvin Calvin discovered the Calvin cycle, kind of like Hans Krebs and the Krebs cycle. But this is photosynthesis. Now, the Melvin Calvin, the Calvin cycle goes by a couple other names. I think I mentioned before. The light independent reactions is a synonym, and the dark reactions sometimes. Trying to think of another one. Light independent reactions, dark reactions, Calvin cycle. Oh, sometimes it's called carbon fixation, because the first step of it is carbon fixation, kind of like the citric acid cycle is named after the first step, which makes citric acid. But anyway, Calvin cycle or light independent reactions or carbon fixation. I won't call it the dark reactions. That's pretty old-fashioned. If you have a professor that says that, then don't tell him I said that. All right, so sometimes it's called C3 photosynthesis, but like I said, we're going to go with Calvin cycle or light independent reactions. The first step of the Calvin cycle is to attach carbon dioxide, which comes from the air, comes from the environment, to a 5-carbon sugar that's already in the cell called ribulose 1,5-biphosphate. And ribulose 1,5-biphosphate goes by the acronym RuBP. RU stands for ribulose. BP is biphosphate. That is really the key step, attaching CO2 from the air to a 5-carbon molecule that's already in the stroma. And this is happening in the stroma of the chloroplast. And it's going to make a 3-carbon, it's going to initially make a 6-carbon molecule, which then splits into 2 3-carbon molecules called phosphoglycerate. All right, but the key thing is to attach CO2 to RuBP. That's carbon fixation, because you're attaching CO2, which is considered inorganic carbon. You're attaching it into an organic molecule that's already in the chloroplast. So that's carbon fixation. And what's critical here is the enzyme that catalyzes this reaction. That enzyme is named, get ready, ribulose bisphosphate carboxylase oxygenase. And I think you can see why we use an acronym. Ribulose, RU, bisphosphate, BIS, and then carboxylase oxygenase, CO. So we call the enzyme Rubisco. All right, the enzyme is abbreviated as Rubisco. So Rubisco catalyzes RuBP plus CO2 yields 2,3-PG. But the key is that enzyme, because it can take CO2 out of the air and attach it and make a sugar from it. So think about that for a minute. Mother Nature made an enzyme that can take CO2 from the air and attach it to an organic compound. And, one thing that scientists think about is, is there a way that we can ramp this up? Is there a way that we can get this enzyme and like manipulate it to pull the excess CO2 out of the atmosphere? Because that's certainly a big part of what's causing climate change. Now, it would have to be a huge idea that they came up with, but scientists are working on this because Mother Nature provided an enzyme that naturally does this. So all we would need to do is somehow ramp it up or increase it or make a lot of it, and maybe we can solve the problems that we have created. Okay, so this is the first step here. This is the carbon fixation is what I'm showing right here. All right, Ruby P plus CO2 yields 2,3-PG. That's also an acronym. But this is the carbon fixation, a 1 carbon molecule attached to a 5 carbon organic compound. Now, there's some other steps, and this is a cycle. And you'll remember when I talked about a cycle in the past, the definition of cycle is that something that you start with is also regenerated by the end. In the Krebs cycle, it was called oxaloacetate. In the Calvin cycle, it's Ruby P. Ruby P is the molecule that's there in the beginning, and Ruby P gets regenerated by the end. With the Calvin cycle, you can't really consider just one turn of the cycle. You have to do it in multiples of 3. So we say 3 turns of the Calvin cycle will incorporate 3 carbon dioxides and produce 1 3-carbon sugar called G3P. And I know G3P is almost the same as 3PG, and they are almost the same, but it's called G3P, glyceraldehyde 3-phosphate. But G3P is good enough, a 3-carbon sugar. 6 turns of the cycle will incorporate 6 carbon dioxides and create the equivalent of 1 molecule of glucose. So we typically think of the Calvin cycle in multiples of 3 or 6. So on this diagram, they show it in multiples of 6. If you have 6 molecules of carbon dioxide and 6 molecules of Ruby P, then each 1 carbon dioxide will attach to 1 Ruby P, but you multiply that times 6. Then when you look at everything that goes on in the cycle, 12 ATP are needed over here, 6 ATP are needed over here. So a total of 18 ATP are needed, and those are provided by the light reactions. Remember, ATP is made in the light reactions, and ATP is going to be used for energy in the Calvin cycle. And remember the NADPH from the light reactions, there's 12 that are needed in this part of the process. So the ATP and the NADPH that were made in the light reactions are broken down in the Calvin cycle, but what we're going to get out of this is 2 molecules of G3P, and each G3P is 3 carbons. And then it doesn't take very many more steps to connect those together. The cell will connect those together and make 6 carbon sugars like glucose. So glucose is not the direct product of the Calvin cycle, G3P is, but 2 G3Ps can be joined together. And other sugars can be made as well. Sucrose is a common one. That's the type of sugar you have in your sugar bowl at home if you have one. So different kinds of sugars can be made from G3P. And then once you have sugars, once you have especially glucose, then those can be joined together to make a polymer called starch, which you'll recall is the polysaccharide found in plants. And so the plant will make sugar, simple sugars, and the plants will then use those simple sugars, connect them together to make large polysaccharides for storage of energy. And my last slide is just to point out that not every plant uses the exact same Calvin cycle. The Calvin cycle I showed you, the one that Melvin Calvin discovered, is the main one. But there are plenty of plants who have adapted the Calvin cycle to particular environmental pressures, and they have evolved different variations of the enzymes needed for both the light independent reactions for the Calvin cycle. And what these variations do, some of them are variations in the enzymes like rubisco. So in some plants, rubisco is not the main enzyme for carbon fixation, or at least it's not the first step of the reaction. But there's different variations, especially for plants that live in the desert, plants that live in areas that don't have a lot of CO2 or water or both. And so these variations that we see in plants are adaptations of the main system that I taught you, but they are adaptations to help the plant survive in really harsh environments. However, very few plants have ever been adapted in this way. Plenty of plants probably found themselves in harsh environments and went extinct, because the way evolution works, it's not an on-demand kind of system. But some plants have adapted to these very harsh environments, and typically what you're thinking of when you think of adaptations are mutations and variations in the basic enzymes. So that helps the plants deal with the extra stress of a desert environment or a low carbon dioxide environment. All right. Okay, so you've got a good overview of photosynthesis, and go back through any of the materials that you might need to rewind and listen to again, get some detailed notes, and then start working on the post quiz.